Hi everyone, my name is Feng, and today I'm going to tell you an interesting story about how the brain and the light meet. So before we go further, here are the learning outcomes. I will first introduce a very important brain functions called the working memory. And then I'm going to tell you how working memory triggers the different brain responses or through the changes of the hemodynamics in the brain through the theory called neurovascular couplings. And then we're going to take a little break and talk about the properties of light. And finally, I will tell you how they both meet. So what is working memory? Working memory is the temporary space that holds information for immediate manipulation. It is extremely important because we use that in our daily life, from language comprehensions, planning reasonings, problem solving to decisions making. And we look at the properties of working memories. It is limited in capacity, volatile, temporary, and requires sustained mental effort and attention to hold the information. So to make it simple, we can imagine working memory as the RAM in our machines. Once power off, information's gone. Now we talk about the theory of neurovascular couplings. Now, even though I believe that our brains is really capable and powerful, but it doesn't mean that we can grow muscle by just thinking about it, right? But the cool fact here is that our brains works pretty much similar like hitting the gym. If you have a very difficult problem set, it's gonna impose a very high working memory load to your brain. And thus, the brain's gonna need more energies and more blood's gonna flow into the vascular brain regions to support the brain activity. And this happens particularly in the prefrontal cortex regions, which is underneath your forehead regions as shown here. Right, so as we say, we'll take a little break and talk about the light. So if I have a light source and the mediums, so what's happening is that the light will pass through, will scatter in the medium, absorbed by the medium, and definitely there will be some traces of light that reflected back to the source. So this is the model of the light propagations. Now I'm going to bring your attention to a particular window that is between the visible red light and before the invisible infrared light. So in these spectrums, it's called the near-infrared spectrums, or NIRS in short. Now, you're gonna ask, why should we care about this particular wavelength? So here are all the cool examples of NIRS in our daily life. Can we imagine going through the pandemic without this cool application of NIRS? Now, we're gonna do a quick test. One of these is actually not the applications of NIRS lights. And I'm gonna count from 3 to 1, and then you're gonna raise your hand, pointing to me, showing with your fingers which numbers that you think is not the application for NIRS. Ready? 3, 2, 1. Awesome. 95% of you showing number 4, which is the correct answer. Congratulations. So number 4 is actually the example of the UVC lights that can use to eliminate viruses on the surface. Now, the key take-home message is that not all the applications that using lights are safe. So UVC is actually harmful to human beings. But other than that, all the NIRS applications that we've seen from the photo here is actually very safe. So now we ask the next question: what can we do with the NIRS and what did it have to do with my brain? Now, to implement NIRS, they call the functional NIRS, we need to have a dual wavelength and place the distance of 3 centimeters between the source and detectors. So if you pay attention to this particular window, we can see that the absorption coefficients for all of the biological properties here are actually very low, which means most light don't absorb. So in this case, you can kind of imagine that your brain sculpt as the transparent tissue here. So all the lights will go through, reaches the cortical regions of the brain, and get reflected back to the sensors. So with that, we can actually estimate the changes of the hemoglobin based on the reflections. Now, to give you a little bit better visualizations, we can see from the photo here. So since we fixed the locations for the source and the sensors during the scanning, we can pretty much assume that the change of the light attenuations between task and resting states is actually due to the change of the hemodynamic response in the brain regions. And that theory is called the neurovascular coupling theory. So if we pay close attention, this is actually a very interesting chart. You can see that the properties of light absorption coefficient between the oxygenated and the deoxygenated hemoglobins actually have a very distinctive patterns, and they changes across wavelength. So in order for fNIRS to work, we simply choose two wavelengths, one before and the other one after the intersections. And with that, we have the 4 points to form two simultaneous equations. And then we can plug in the model called the Beard-Lambert law. They're trying to translate the changes of the light attenuations to changes of the brain hemodynamic response. So if you feel overwhelmed, the red plot here simply means a hemoglobin with a friend called oxygen, and the blue one is kind of lonely without a friend. So one cool application for fNIRS is that we can use it to monitor the brain activity continuously. So in this case, we can check how much is the participant engaged with the task and subsequently adjust the workload. So with that, we can maximize the training effects to promote cognitive learnings and also to prevent dementia. Now, we come here to ask the last questions. Out of so many neuroimaging modalities, why should we use fNIRS? So in comparison to fMRI, the gold standard, fNIRS doesn't require a huge MANEC, and thus it makes fNIRS more portable, support naturality settings, relatively cheap, and it is highly correlated with fMRI both signal. And fNIRS also easily set up, and thus it is very suitable for studies that involve infants. Of course, there are also limitations with fNIRS, which is the lower spatial resolutions, and it doesn't go to the deeper brain region. Now, to give you a quick view of comparison between fNIRS and fMRI. So you can clearly see that if we plot the activation map based on fNIRS, it is more pixelated as compared to the fMRI with superior spatial resolutions. However, there are two advantages for fNIRS, which is higher in temporal resolutions, and it provides an additional information about the deoxygenated hemoglobins, thanks to the two wavelengths. So in conclusion, it all depends on your applications. If you're going for a detailed scannings and going for a deeper brain regions, fMRI is the right choice. However, if you're really curious and study the brain activity during a more naturalistic settings, for example here, learning or infants, neurodevelopment, or the mother-child connections, or even outdoor activity, fNIRS is definitely most coolest modality that you should consider. And with that, I thank you for your attention.